Alright, so this is our lab 2, which is just one exercise in your book, and as you've already experienced, it's a lot of stuff though, right? So we get this very own lab. And so it's examination of eukaryotic microbes. So all these organisms we looked at today are all eukaryotic. So there's several terms that you were supposed to go through, and as always, this PowerPoint will be posted for you guys after class, so you can't cheat, right, on your homework. Sorry. Okay, but I'm not even going to go through all these terms, but it's always good to kind of have a glossary, right? Read over your terms and be familiar with them, and I may have slightly definitions that help you understand these terms better. And I'm never going to make you write out terms on a test or a quiz, but based on a definition, I expect you to be able to identify a term. Terms are another thing that you do need to be able to spell for your practical exam and sometimes on quizzes. One of the things that if spelling's an issue, may be one of those things you need to practice. So I'm just going to actually scroll through these real quick, because I don't have enough time to go through all of them. So we start out talking about different cell types. Right? So we have prokaryotes versus eukaryotes, and as I already said, this lab is all eukaryotes. So all of them, at one time or another, or if they're stained properly, and depending on their size, for a lot of you were actually able to see their nucleus, and their nucleus actually would help you identify. The nucleus is a membrane-bound organelle. It's actually double membrane, and it's a defining characteristic of this cell type. In addition, these cells will have other membrane-bound organelles. This is organization within the cell. So, for instance, Spirogyra, which is an algae, what did you see? Nope. The Spirogyra has like spirals in it, but what are those spirals? Chloroplasts. They're chloroplasts. They're chloroplasts. So, prokaryotes, which is what the rest of this course will be, unfortunately, to a certain extent. But bacteria are pretty cool, and they fall under this cell type of prokaryotes. And so that's pretty much what we'll be doing the rest of this course, and they're teeny tiny, and you will live under oil immersion, right? So getting that technique down is very important. If you're scared of microscopes, you don't do well with microscopes. The study lab has microscopes. When this lab is open, when I have office hours, if you want to spend extra time looking under the microscope, come see me. It's my favorite thing to do, okay? I want you guys to be comfortable. Because you're going to need to focus your own slides, right, and look at your own stuff that you make. So I want you to become proficient, and I'll teach you all the tips and tricks of the trade, right? So prokaryotes, by definition, pro before karyote actually refers to the fact that for beginning scientists, the nucleus almost looks like a kernel inside the cell. These guys lack that. It doesn't mean they lack DNA or any of the things else that would be in a nucleus. It's just not membrane bound, right? It doesn't have that level of organization within the cell that eukaryotes do. So here you see a sample they took from the gum line, and you see a big flat eukaryotic cell. They actually gram stain this, and we come out gram negative because we don't have cell walls. And there's the nucleus. There's actually two cells. Here's another one overlapping probably underneath this one. You can see the nucleus. Those are our cells, our eukaryotic cells from our mouth. And then you have a bunch of gram-positively stained bacteria, all these purple dots that you see. So the stuff we were looking at today are very big in comparison to bacteria. Yeasts were tiny today under oil. They're actually bigger than bacteria. Right? So what we're going to be looking at later is even smaller than the smallest stuff we looked at today. So, classification, organizing stuff, right, into these different groups that we say, like algae and animals and protozoa and fungi. That was begun by botanist Carol Linnaeus, right, way back when, 1958. And back then, right, he was just really making the distinction between animals and plants. Right? We know from just looking at today, you know, it's like, well, is an algae a plant? Kind of is, right? But kind of isn't. Is a protozoa an animal? It's a single-celled organism that eats other single-celled organisms, right? So we have a lot of these differences that we had to have much more categories as we moved on, right? As we discovered more and more, especially once we get into the microscopic world, which even then it can be quite interesting. So you probably learned the classification scheme, right, of the groupings, the common groupings that we do for organisms, and for the most part, how are these groups determined? Like, why are all the bears in the same family, right? Not actually genetics. In the beginning, it was just basically traits, what they look like, what they do, right? So in the case of bears, they all kind of look the same, right? And then when you get into the smaller groupings down to an individual species, and then as we travel up, carnivores, right? These are all meat eaters, okay? Mammals, they have the characteristics of mammary glands and hair, right? And when we get up to chordata, they all have the similar feature of having a spinal cord, okay? But notice, you know, we've got things like snakes that definitely aren't anything like a bear, right? But this is a very large grouping, right? Based on that key characteristic, that type. And then animals, right? You even have things like sea stars at the kingdom level. And even above that now, especially in the microbial world, we have what's called supergroups, right? So you've seen that, like, they've kind of gotten away from kingdoms, and they have these supergroups and subgroups, right? So that's somewhere in this area of classification. And then above that, probably something you haven't talked about before, are domains. And this is where we really started actually looking at genetic type information. In this case, ribosomal RNA. And it was Carl Woese who came up with this scheme in the 1970s, 1977. And he grouped organisms into 3 major domains based on their ribosomal RNA, right? The ribosomes that make proteins. Every cell have these structures. Every cell has to have this ability. So we ended up with eukarya, which is what we're studying today, and it comprises of all eukaryotic type cells, hence the name eukarya. When we get to prokaryotes, though, we actually have 2 divisions. We have bacteria, which is where we'll live in this class, and we have archaea. And these guys, although prokaryotic, don't have a nucleus, they're really small, they are quite distinct and different from each other. And again, very few, I think they have like 1 pathogenic archaea they've learned about. These are organisms that tend to live in really extreme environments, extremely cold, extremely hot, extremely salty, things like that. So, of course, we're not in an extreme environment, so they wouldn't necessarily be pathogens to us, make us sick. So, of course, bacteria is a major focus for humans because of the bad things they cause, but they're also awesome, good things. We wouldn't survive without them, right? And we certainly wouldn't have beer and wine and cheese and cool stuff like that if it wasn't for microbial fermentation. So, and then, of course, this is just for life. And so viruses don't factor into this. Most scientists don't consider them living. Right? So this is the organization of life. So again, traits, but then we also are adding in more information like ribosomal sequencing to help delineate and group these organisms together. So, what domain are we looking at today? Who's been paying attention? Eukaryotes. Eukarya, right? So, eukarya today, all about eukaryotes. So, this is a mnemonic that I learned when I was in school back in the day of remembering kingdom, phylum, class, order, family, right? Which you don't need to memorize for this class, but maybe for other classes. So it was Kevin, please close our front gate slowly. Peter, one of the other professors that I co-teach with, learned King Philip came over from Germany, right? That was his little mnemonic saying, right, to help remember kingdom, phylum. But we really need to add a letter, right, because we have domains above kingdoms. So my sentence is very easy to add the D, because you can just say, damn, Kevin, close our front gate slowly, right? D for domain. So, not to be confused with mnemonic plague, right, which is a whole other thing, right? A mnemonic is a device for remembering things, right? And a lot of people use mnemonics. I used to my very energetic monkey just slipped under 9 pies. That's the order of the planets from the Sun to Pluto when and if they decide it's a planet and then change their mind again. So taxonomy is classification of the organisms into categories. Phylogeny is a little bit different. This is where we're studying those evolutionary relationships, right? So who potentially evolved from who? Ontology is the study of development of an individual. So even in our development, right, we have a tail, right? We lose it for the most part. It doesn't actually stick out of our body, but we all are born with and have, we all develop with a tail. We're not born with it, right? Although some people could potentially have tails. So ontology replicates phylogeny. May or may not true, right? So this developmental process may give us clues into these evolutionary relationships, right? When you even look at the developmental phases of how an organism develops. So just looking at characteristics, right, can really kind of lead you astray if you're trying to develop evolutionary relationships, you know, what came from where and who was whose ancestors, especially when you look at something like these cute little whales and fish, because they have the similarity. If you just look at the fact that they live in the water, they have fins and can swim, you could kind of group them all together. But then when you look a little bit deeper, right, we know that fish have scales, where whales are actually mammals, right, and they have hair. And whales have live birth, where fish lay eggs, right. Fish can actually breathe oxygen from and acquire oxygen through their gills through the water, where whales actually have to go to the surface and get atmospheric oxygen in order to breathe. And then sharks are a whole nother beast, right? They don't actually have bones like fish do. They're actually made of cartilage, right? Softer tissue than bone. It's not calcified. So it's really easy for humans, especially if you kept track, right, of who your relatives are, and genetically, right, you look the way you do because of the traits that you inherited from your parents. And so this is just a tree I found online, right, and you can see that, you know, these two guys are related by this common ancestor, which would be their great-great-grandparent, their third cousins, right, 3 generations down the line. One of my favorite commercials on TV now is the Ancestry.com, and the guy thought he was German, and then he had his DNA tested, and it turns out he's Scot-Irish, or something like that. And it's one of those things where, like, there was some misinformation at some point in the family, right? And they thought they were, you know, but it might have been one of those things where, you know, they went from one country to another, not to say that they were of that nationality, right? I'm a 100% mutt. You probably name any European, right, including Indians, right? I have it like, my background is all over the place. I'm a true mutt. I usually say, just name something. Yep, I got a relative that came from there. So when we are studying these organisms, when we look at these relationships, there's some interesting theories out there as to what may have happened in the past. The belief is that we probably started out with mostly prokaryotic organisms. They hadn't gone to that level of organization of having a nucleus. And so, but you had this distinction, and they probably branched off from a primary prokaryote, but they kind of went their separate ways. You had ones that evolved into what we know nowadays as bacteria, and others that evolved into archaea. But some of them even crossed back over, right? And there's this interesting evidence that we have about some of the organelles, right? When you jump this line from prokaryotic to eukaryotic, the organelles like the chloroplasts and mitochondria, those organelles have their own DNA, they have their own RNA, and the RNA, the ribosomal RNA, part of the ribosomes that they have inside there, is of prokaryotic nature. It's not eukaryotic. So there's this belief, this endosymbiotic theory, that one prokaryote engulfed another prokaryote, and they began this symbiotic relationship so much so they couldn't separate from each other. So what became eukaryotes was probably from a combination of two prokaryotes coming together, right? And the continual evolution there. So you have this mid-region of protista that really represent this mixing that probably happened. And then you have that branching off where they really went more towards being plants, right? Where some protists down here, they can photosynthesize and they can eat, right? Where plants, you're just talking about photosynthesis for the most part, except for like Venus flytrap that eats, you know, flies. But it can still photosynthesize. You know, have exceptions to the rules. And then you have others that branched off, like fungi that became our major decomposers, our saprophytes that eat dead stuff and put it back into the environment. And then you have our branch of animals, right, where we primarily eat plants and animals for our nutrition. We can't photosynthesize, right? So really cool stuff, right? And as we study the different components of cells and make these different relationships between things. So here are the three major branches that I talked about, these domains that Carl Woese and apparently George Fox came up with. And so you have the major portion, bacteria, right, which we'll focus mostly on in this course, your other prokaryotes, your archaea, and then what today's lab focuses on, which is eukaryotes or eukarya or eucota. So we're humans, or eukaryotes, right? And we would be under animals, yes. We forget sometimes, right? We're in that grouping. Put ourselves on this little pedestal, like we're above animals. No, we really are animals. So remember I said about these supergroups, these really big groups? Some texts will have 5 groups like our book. Other places, like where I stole this pretty tree from because I wanted a tree with more branches than they had in the book, actually has 6. And where the Euglenozoans, they split into these 2 additional supergroups, right? So it's either these and this one for 5 or 6, splitting this one into 2. Notice we've shrunk down the bacteria and archaea because we're focusing on eukaryotes. So we've expanded upon those relationships. So on this one, where the heck would humans be? We're animals even, right? So this weird name you may have never heard before. This is Metrozoa, right? And so all these links and stuff in my PowerPoint were working so great, so I have a whole page in Canvas for you guys. So Berkeley, it's Berkeley, yep, has an awesome website, like a virtual online zoo of sorts of the fossil record all the way through for animals. So if that's something that interests you, there's that really cool reference for you. So prokaryotes versus eukaryotes, right? So for prokaryotes, what are our 2 domains? Bacteria and archaea, right? And this is right straight from your homework, right? Eukaryotes are just one domain, and that is eukarya. Right? Some texts will have kingdoms of plants, animals, protista, and fungi. This is microbiology class, so of course we get rid of the first 2 because they're not microscopic. So we're going to focus on protista and fungi. And again, these are kind of unofficial groupings. So then, right, we have these supergroups now, which by no means do I expect you guys to memorize, right? You're not going into taxonomy. This is not something you need to know. You need to be aware of, though, right? Classification. One of the things that will help you that probably is important because these terms are used pretty widely is lumping protista, right, protozoas that we've looked at, amoeba, right, things like that, along with our algae like the Spirogyra and the Volvox. For fungi, right, a common grouping that's used, we talk about yeasts, which are unicellular fungi, as opposed to multicellular filamentous fungi we know as molds, right? Most of us have seen mold, unfortunately. And then you have very large macroscopic fungi. Again, they're macroscopic, so we don't talk about in microbiology. but good old mushrooms, right? Things such as that. So we're going to start out on one of the supergroups, and we're going to look at a couple of representatives. So we're going to start in Excavata, and one of the subgroups that we're going to look at has this particular organism, which unfortunately we don't have slides for yet, but I'm hoping to order some. The good news for you guys, because you don't have to memorize this one. Yay. And then this guy we don't have good slides of, but he is in your book, right? And we did try to get slides at one time, so Giardia you do need to know. But he's kind of cute, right, with his little paired nuclei. Almost looks like he has eyes. And we don't have any Euglena, but again, my hope is that we will in the future. This is one, if you look at pond water, you usually see sometimes. So you may have seen this guy before. So what does this group have in common? They're all unicellular. They all have a feeding groove where they're going to bring food into their bodies. They'll tend to have one or more flagella. And most of them will have a trophozoite and a cyst phase to their life cycle. The trophozoite is the feeding phase. The cyst is the resting state. This tends to be for pathogens, the one we would ingest, and then it would come out and form the trophozoites and make us very sick. It's definitely the case when it comes to Giardia. So Trichomonas, this one I've seen videos and stuff on. This is really cool because it has like this undulating membrane, or it looks like it's waving. It's really cool. In flagella, right, distinctive nucleus. Only the trophozoite's known. It's not known to form cysts. It's a fermenter, which is really interesting. We're going to talk a lot about fermentation and how organisms acquire food and stuff in this class. And this is another one where, you know, it has this ability that's usually more of a prokaryotic ability of being able to produce hydrogen gas under anaerobic conditions. So it's actually using hydrogen as an energy source. So what sexually transmitted disease is caused by this organism? Trichomoniasis, which is a sexually transmitted disease, right, STD. Not to be confused with other trichinosis, right, or trichinella, right, which is a whipworm and or a roundworm. Most of the time, it's going to cause an infection of the vulva, right, the lips outside of the vagina for women. But men can be carriers as well, although thankfully for them, their reproductive environment's different pH level, and they don't tend to like that so much. Women are at a slight disadvantage, environmentally different. Giardia labia, and again, this one was named after it because it sometimes infected the labia, the lips, the vulva to the vagina. But we've come up to find out that it is, in fact, just one species, the same species that will also infect the intestines, and some people even have it and aren't even sick. So they've most texts have agreed upon, and most scientific literature, you won't see labia anymore. You'll see instantalis for the intestinal. But your book still has labia. So can you see the 4 paired flagella in this picture? No, it's a crappy picture in our book. Here's a drawing that actually shows the flagella, and then we have some pictures, a poster of one of my students had done in the packs, and some information on Giardia up in the back of the room. This is the trophozoite. It does form the cyst, and that would be the one that you ingest and it would make you sick. These guys have metrosomes, which are non-functioning mitochondria. They don't function. So they're stuck just doing glycolysis. They can't do electron transport chain. So as you can imagine, they really need other organisms to help them out. They still do glycolysis, though. They have paired nuclei that almost look like eyes, right? But they are the nucleus. They just actually have 2 within their single cell. They have the 4 paired flagella. So actually have 8 total. 2 medial bodies. And you can see them right here. Sometimes in some of the pictures they almost look like a smiley face. The cyst says what typically you're going to ingest, and it would be infectious for us, right? And it resists most forms of treatment. But again, our water supply, we usually keep it out of it, right? And we test for things like that. Don't even get me started on that with the amoeba right now, right? So you'll notice on some of these slides there's a bunch of information that I'm not going to read, right? It's extra information, especially a lot of you guys are nursing, allied health majors, right? This is good informational stuff for you, but I don't have enough time to cover it in lecture. It's not anything you'll be tested on either, right? It's cool stuff. Okay, so for Euglena, these guys are green because they actually have chloroplast. They photosynthesize. They have 1 or 2 flagella. They have an eye spot, also referred to as stigma, and this allows them to sense where the light is, and of course the flagella allow them to be modal through the water so they can head towards light to be able to photosynthesize. But they also can engulf food. So they're autotrophic in that they can make their own food, they make their own carbon, or they're heterotrophic when light isn't present, they can eat other organisms to attain nutrients and carbon that way. So, you know, this is one of those ones like, is it an animal or is it a plant? Kind of acts like both, doesn't it? Right? It's its own little entity. So, what structure gives these organisms their photosynthetic ability? They're chloroplasts. And hence why they're green, because they have chlorophyll. Trypanosoma, I wish we had this one, but we don't. Hopefully in the future we will. This one is transmitted by the bite of a tsetse fly in Africa. If it is the species Brucei, and there's actually some subspecies, and I have a whole other chart that Peter put together for you guys that are interested in pathogens and stuff, and it lists all the pathogenic ones from this lab, and it's a cool chart that has what transmits them, you know, what diseases they cause, and cool stuff like that, all organized in one spot. So this one they just talk about in your book, sleeping sickness, which this is in Africa, where they have the tsetse fly. They have this protozoa that, I mean, yeah, this one would be considered a protozoa, that's transmitted by the bite of that insect. And it can cause really serious degeneration to the meninges surrounding the brain and spinal cord. So eventually the people, you know, they can't function anymore. They go to sleep and die. Right, so that's why it's called African sleeping sickness. There's 2 different species, subspecies of Brucei. Gambesi is the one that mainly infects people, but there's one that can infect people that actually comes from cattle, and that's Rodentia, I believe. But there's another Trypanosoma cruzi that causes a totally different disease, transmitted by a totally different vector, and a totally different part of the world. So our next grouping is where you get into what's more formally known as algae and more closely related to plants. So we have our chlorophytes, and this is actually, I don't remember if that's, yeah, I think this is the Chlamydomonas. Yeah. And we don't have these guys. But one of my YouTube videos, you can see them swimming around, but you can't see them in this detail. They commonly come in our live specimens, and hopefully I'll have for you guys next week to take a gander at, in with the amoeba. They usually have it as a food source for the amoeba. And then we have Volvox, which you guys did see today. And unfortunately, you can't see the individual cells. Like, if we had the ability, right, these specimens are so thick that even if you try to go to oil immersion, you're not going to be able to see in this level of detail. You're not going to be able to focus in on the single cells. Even then, notice they were only able to focus in on a couple of these. The rest are out of the plane of focus of that microscope. So these are colonial. They're actually made up of little tiny cells that live in a colony. And then this is a smaller colony within a larger colony, and they're called daughter colonies. So then another group that we have has the Spirogyra, and you can clearly see these. This is much longer filamentous one, where you can see the chloroplasts, and they actually arrange in spirals, which makes it really easy to remember the name of this one, right? Spirogyra. So what do all these guys have in common? They all have chloroplasts, all autotrophic, they can make their own food. And they contain cell walls made of cellulose, which is similar to plants. Plants, their cell walls are made of cellulose. So chlorophytes you'd find in fresh water. They're unicellular, filamentous, or colonial. And as you can see, the Chlamydomonas is unicellular. And Volvox was colonial, right? Lots of cells living in a colony. Chlorophytes is filamentous, and this grouping, Spirogyra, is our closest relative, probably to plants. So Chlamydomonas is pretty cool. This is a zoomed-in picture. It's haploid, which means it has half of its chromosomal information. It's unicellular, single cell. Has a unique chloroplast, this particular shape to it. Has 2 flagella. And a stigmata, again, so that it can sense where light is and travel to it. There's a really cool video Peter found for me. I used to just have the song of a professor did of Chlamydomonas. Those of us that are nerds love it. I don't have enough time to play it in class, so check it out if you're interested. He's singing with his guitar about Chlamydomonas. It's got some cute little jokes in there. Not to be confused with other things that sound similar, right? Chlamydia is completely different from Chlamydomonas, right? It's a bacterial infection. So we saw our Volvox that are colonial, right? They can reproduce sexually. Their whole colony would then be all sperm bundles or all eggs, and then they would travel and mate with others, and then they would produce a whole nother colony. Or they can reproduce asexually, where they just split, right? And when they do asexual, they form daughter colonies within the colony. So this is a colony, and it would eventually leave a colony. And you can see in this picture, though, this is a colony that's deteriorating, and these were its daughters that even have daughters themselves, right? So they just keep dividing. So that's asexual. The daughter colonies that you see within a colony, the smaller colonies are daughters. And that's asexual reproduction. So, really cool website, lots of pictures, lots of information. If you're interested in these organisms, they're pretty cool. Spirogyra, they're filamentous, right? So they're long filaments. They're haploid as well, single set of chromosomes. They can form conjugation, where they'll exchange genetic information and form a sexual spore that will then go through meiosis and produce more haploid, but different from the 2 parents, right? That's a form of sexual reproduction. And so we actually have some of those slides now, but we didn't have them in the past, and they do show this in your book. So there was one slide today that showed that conjugation, not responsible for it for testing and quizzing purposes at this time. So what you do need to be able to identify is the chloroplast, right? You can see a nucleus, and you can see the cell wall that divides the 2 cells within the filaments. Our next grouping has the really cool paramecium, right? Things that you've probably hopefully seen in biology class previously, and hopefully we'll have some live guys swimming around. If not, YouTube has got a great resource, and I've picked out a couple of awesome videos that really show these guys in beautiful detail. Centaur is another one of my favorites, but we don't have right now. And this is an awesome one to watch, live specimens swimming around. This really cool trumpet shape it has to it. The pathogenic one we have is Bellatinium coli, and this one we did have. And you notice it has that characteristic nucleus shape that helps you in identifying it. It's actually the only known ciliate one that's covered in cilia that causes infection in humans. Then we have our beautiful diatoms that fall into this same grouping, although sometimes they're referred to as algae. And this is actually a protozoa ingesting some diatoms. And so you can see there's lots of different shapes out there, but we base them based on their symmetry. So the kind of circular ones are radial symmetry, or referred to as centric. And the ones that are elongated or bilateral, side to side, are referred to as pennate. This is an aside that I'm not going to go over, but it's cool stuff you can read about. This is my little pictorial representation of endosymbiosis, where one cell engulfed another. And they believe that this even happened a second time, where you have an algal cell, right, that engulfed cyanobacteria. These guys were photosynthetic. Now this cell was photosynthetic. This probably became the chloroplast. But then even algal cells were once engulfed by another cell, and it was actually red algal cells, to give rise to this particular grouping that we're looking at now. So they have these chloroplasts that were derived from engulfing another eukaryotic cell. So for these guys, we have Paramecium, Centaur, Bacillium coli. These guys are all motile by cilia and have an oral groove and a mouth, a cytosome, where they move the food in and then put it into a vacuole and digest it within. These non-motile ones, the Plasmodium, these have very complicated life cycles, and they're usually parasitic. And in this case, Plasmodium causes what disease? Do you guys know from your homework? Malaria. And then our subgroup, most of them have a unique flagella, but diatoms do not. But they're so cool and highly prevalent, that's why they were picked as a represent for the group, even though they're not a good representation. They don't have the key feature of the group. So for ciliates, for the Paramecium, we don't quite have a side quite as beautiful as this one. We don't have anywhere the cilia are stained. You can just see the general shape of the organism, and you can at least see the macronucleus. I haven't found anywhere you could see the micronucleus. But you do need to know about their features, right? That they have cilia, they have an oral groove. In this electron micrograph, you can see the oral groove. The contractile vacuoles, right? Definitely check out that video, and hopefully if we get some life specimens, we might actually get to see it in action next time. But of course, on a test, I wouldn't necessarily ask you to identify it because we don't have slides that have it. You have to see that stuff live. So I have these links in Connect for you. So Centaur is another one that unfortunately we don't have. Like I said, it has this cool trumpet shape and beaded macronucleus. And you can see within here, that's where its mouth is technically, its cytosome. They're naturally green. They're really beautiful. These guys are also naturally this color as well. These are the parasitic ones you do need to know, and we did look at under the microscope, and really the key feature is that horseshoe-shaped nucleus. And this is the trophocyte. They do form the cyst, which is usually an infectious phase. Plasmodium. We had red blood cells on our slide. Our slide actually has, they're pink. This one's been stained, so the red blood cells are kind of bluish in this picture. The parasites in this picture are actually inside the red blood cells, and you'll see them inside and outside the red blood cells, right? Because again, complicated life cycle. So you need to be able to identify the parasite, right? That you're looking at the parasite and as opposed to the red blood cells. And so we said this one causes malaria. And this is a serious disease in other parts of the world transmitted by mosquitoes. Thankfully, we don't have the protozoa here, but God, do we have mosquitoes, right? And at one time, they did have malaria in Louisiana, right? A long time ago. The good news is that the protozoa hasn't been able to persist in our environment. We don't have enough environmental places for it. So, lots of different shapes to the organism and several different components of the life cycle. So the good news for you guys is just recognizing that you're looking at the parasite and whether it's inside or outside the red blood cells and not the different phases of the life cycle. That would be crazy. Right? But these are all the different morphologies you would potentially see. In humans, it also infects our liver. But the easiest place to get a sample is the blood, because it also infects red blood cells. So it's quite damaging, causes lots of problems worldwide. Other bad news, guys, right? Mosquitoes, as we know, carry other things, right? Things that we even have here in the United States, like West Nile, and yellow fever at one time, but not anymore. Most of the times people are getting these from other parts of the world where, again, the viruses that cause these diseases are present and can be transmitted by the mosquito. And then ticks are bad news too. Right? And on the door coming into lab, you'll notice information about ticks and mosquitoes and fleas and all that good stuff. So distribution-wise, right, where you have to worry about, and this is just one of the 4 species that cause malaria, and this is the worst one, falciparum. But there are actually 4 different species. So, interesting thing about the prevalence of sickle cell is related to malaria, because sickle cell disease, because it affects the red blood cells, it affects the ability of the protozoa to infect this Plasmodium, to infect the red blood cells. So people who have partial sickle cell disease, or what we call being heterozygous, only partial trait, only partial sickle cells, are much more resistant to malaria. And so therefore that trait has continued, and mostly in the African American community. And the unfortunate thing is that if you inherit both of the defective traits and you get full-blown sickle cell anemia, it's worse than malaria. Right? And there's the chance of that because of the high prevalence of that genetic defect, because it's beneficial in that environment. Right? So we select for it. We have that selective natural pressure for it. So, and this is, so as you can see, you know, sickle cell goes along with the regions of malaria because it's somewhat protective. So that brings us to our diatoms. These are my favorite. These are sometimes referred to as nature stained glass because they contain a substance in their cell walls that actually glass is made out of. I'm going to speed up to that one. It is silica, or silicon dioxide. And so, especially with special types of microscopes and different refractions of light, these guys literally look like stained glass. They're just absolutely beautiful. And, you know, we've got some pretty ones on our slides. And again, we're looking for the 2 different types of symmetry, being able to identify the centric forms, which ours on our microscope look more like this, and the elongated pinnate forms. These are single-celled or colonial organisms, autotrophs, right? They can photosynthesize, right? They've evolved from red algae. They actually have kind of a brown colorization to them normally. They actually have 2 halves if you're able to look at them, and they overlap like a petri dish does with its lid. They don't fit perfectly. Euglenozoans, so this is going to be the closest to us, right? So we're going to be looking at amoeba-like organisms. So this is your entamoeba, right, the pathogenic one. Here's a good old amoeba. Here's actually a drawing, but I thought it was so impressive, of an amoeba eating a paramecium. So Amoeba proteus is the species we have, right, but we're just focusing on the genus. And within the genus of amoeba, they're all pretty much the same, right? And hopefully we'll have live ones to look at next week. These live in fresh water. They're free-living. They have a nucleus, which is seen prevalent on all our slides. And they move by pseudopods, right? No cell wall, because otherwise they wouldn't be able to morph shape like they do. So you can't look for a particular shape for these guys, right? More like a splat. So they move by pushing their cytoplasm. So I've got a cool video that we took in lab, and you can see the Chlamydomonas swimming around in this one, as well as some other ones on the internet where you can see it actually using its pseudopods to surround 2 amoeba and engulf them. So Entamoeba histolytica, unfortunately we don't have any good slides of this entamoeba. The trophocyte, right, slides are poor condition. But if you really want to challenge, right, I got a couple slides where I know I found them, right? If you want to really see if you're good at oil immersion, we'll see if you can find it. Me and Sarah Brock raced one day to see and try and find them on slides, and I won. But we've got 20 plus years of experience each looking under the microscope and manipulating the microscope, and it took us a good little bit of time to find. And I circled even on the slide as best I could the area in which I found it. And some days I can find it again, others I cannot. So certainly weren't going to make you guys struggle to that level trying to find them on these slides. And they're also fecal samples, so you got other lots of crud to look at, and you really got to know what you're looking for. And knowing what you're looking for when it comes to entamoeba is you'll notice this very distinct nucleus, right? It forms this almost like a bullseye structure to it. And so I think this is one of the answers you didn't actually have in your book, right? But it did say it causes dysentery, right? So how are you going to get a sample from somebody? You're going to take a poop sample, right? A stool or fecal sample. And so they don't typically have a characteristic shape, right? They're amoeboid. Fungi. So here's some looking at some yeast where you can really see that budding, that asexual reproduction. And then sometimes these buds will elongate into what's called pseudohyphae. And definitely under different conditions, the Cnidia one, which can be pathogenic, an opportunistic pathogen for us, the Cnidia, it has this characteristic. So that was one of the things I wanted you guys to look for and see. And I'll help you distinguish the two molds that the two yeasts that we look at. So as far as fungi go, right, they're non-motile. They have cell walls that contain chitin, which is a polysaccharide different from cellulose. So they're unique. They're absorptive. They actually release enzymes into the environment, right? So when they're growing on your bread and in your cheese, they're releasing enzymes in, and then they're digesting the food, and then they're absorbing it. Yeasts are unicellular. They're vegetative, actively metabolizing cells, and then when they're reproducing, they have the little buds on them. And this is that uneven cell division you'll see. So here's a good example of this was the first bud that it produced, and then now it's producing even another bud. This is pseudohyphae, right, where they've elongated. Not to be confused with the filaments that you see microscopically for molds. Right? The individual threads that you see here are hyphae. That whole big mass you would see growing like an agar plate is referred to as mycelium. So all of this, right, all these threads, that's hyphae. That's a mold. Right? These are yeast, and you can see the budding here. So we had two yeasts that we looked at, and we had three different molds we looked at, but one of them we have both the asexual spores as well as the sexual spores. So for Saccharomyces, although I think the label in your book says that this is, and they look similar, right? The difference here with Saccharomyces is the specimens we have and the way this organism grows, it doesn't form those pseudohyphae. So how you differentiate them is the Cnidia will have those pseudohyphae, right? They'll have those long threads, right? So these are kind of oval shaped. If you see the buds, right, they're reproducing asexually. If you don't see a bud, right, it's just a vegetative cell. In some of them too, you can see an area, right, where you can see the nucleus. Does this yeast cause disease in humans? No, this is the friendly one. This is the one we like. This is baker's yeast, right? Or brewer's yeast. This is the stuff that makes bread and beer and wine. This won't make you sick. Unlike Cnidia albicans, this one, right, produces budding cells under certain temperatures, but when it grows in our body or on our body, within our body or on our body, it's going to produce those pseudohyphae at the warmer temperature. So you'll see those long threads. That'll clue you in that that's Cnidia. Canada. I keep wanting to say Cnidia. It's Canada. This one can be a pathogen. It is part of our normal flora for most people. You have it on your skin. For women, it's associated with the vagina. It usually only overgrows when there's an imbalance. So for women, sometimes that's after you've taken antibiotics and killed off your good bacteria that keep the yeast in check. right? Or hormonal changes. Or for diabetics, the fact that you have way too much sugar and you overfeed your yeast and you get yeast infections. You can get them in your mouth, right? Immunocompromised individuals have issues with that sometimes. Again, it's an imbalance in your microbial environment, allows these to overgrow, and it becomes very discomforting. Very obese people that have skin folds, right? In those skin folds, because again, moist environment, warm, right, will create an environment that's favorable for those types of infections to grow, right? To overgrow and become infectious. So molds, pretty. I think these are some of the prettiest slides we have, right? So these will grow actually in the bread, right? And then they'll also grow up out of the food source, like even on our agar plates. And this is where they're producing the aerial hyphae, sometimes referred to as sporangia. And these are going to form the spores. For Rhizopus, we have the asexual, as well as if two hyphae come together, like in this bread, two molds come together that aren't the same, they can mate with each other and exchange genetic information and produce a sexual spore. That's your zygospore, or zygosporangia. And then, of course, the thin filaments that make up the organism, right, that you see under the microscope are the hyphae. So we had 3, and you can really differentiate one from the other because each one of these different genuses form their spore structures differently, right? So you can't really see it in this picture, but my other one, you see like a cap on this one. This one, it's a round structure, right, and lines of spores stick out on it. In our book, you actually have a cross-section of it. Those are very expensive slides. I've actually ordered some. They're like $10 a piece. So we'll actually have them sometime soon. So there's no confusion between the book and what our slides look like. I'll have both. And then for penicillium, this one's really cool, right? Their structure where their spores form, it looks like a brush, and that's actually what penicillium means. It means paintbrush. So again, mycelium, that mass that you would see growing, say on an agar plate or on your bread. And then this is microscopically under the microscope. This part was growing in the agar, this dark portion that I stained, but all the little threads, right, are the hyphae. And then the lighter section up here is where it's growing up out of the agar. Right? And you can see the threads, the hyphae, growing up out of the agar. So Aspergillus probably is the best one too because it's got really nice mass of hyphae that you can see, right? And then again, we're looking for the sexual spores. Like I said, we don't have slides that actually have this in cross-section. It's pretty expensive, but I've ordered some. So what commercial products are generated by this genus of organism? Citric acid. And it's, some species are used to make soy paste and soy sauce from soybeans. Penicillium, we know this one for making penicillin, right? But you can use it to make other things too, right? Cheese, right? Some of your blue vein cheeses actually have this, and it's not producing penicillin, the antibiotic, under these conditions. So people who are allergic to penicillin can eat this stuff. I didn't do it, huh? I know, I tried. Last one. So Rhizopus, this one is commonly found on your bread. This wasn't in your book either. This is probably one you might not have got. And again, we have the 2 different forms. We have the asexual, where you can see the spores in this cap, and then the sexual, where the 2 come together and form that zygospore. And this just shows that life cycle. And then, as I said, on Canvas is that cool document. And don't forget that practice quiz. And I'm going to do like I did for you guys before in the announcements. I'm going to kind of give you a checklist. Keep you on task. And this will be posted.